One second. Yeah. Where we are starving without water. We have drought in most of the conditions, and water and world pure water or edible water is rapidly reducing. Even though our earth has so much water, not all of the water is consumable by human beings. Okay, we'll have a technology to change the oceans to fresh water. Yes, you can. But are we leaving the oceans alone? No, everything is polluted. So now, when it comes to scarcity of water and the water consumption by human beings, it is quite shocking, right? So, but this equation is not matching. Understood? So we are scarce and we are using so much water. If you go to bigger cities, the amount of water being used is, I don't know, it's like really a very large number of things. Take mega cities like Delhi, Mumbai, Bangalore, Hyderabad. You take any big cities. Do we have less population? Imagine how much amount of water you use every single day in your own hostels or in your own house. Okay, so it's extravagantly very, very high. So now what happens that leads to scarcity? Now, if we ignore all these utilities that we do, thinking that, okay, I've used the water, so now I need the fresh water for the repurposing my needs. So is it possible for you to keep on going like this without having the sustainable approach? It's not possible. So that is why what we have to do is we have to choose water sewage or sewage treatment of water so that we can recycle, reuse, and make sure that whatever we are polluting is at least not toxic to the environment. So what happens is we have a lot of municipal water that is being produced by humans, which is contaminated of so many things. Now, if you just see a drain, the drain is not just containing only your organic waste. It contains inorganic waste, it contains chemicals, it contains all these microorganisms, it contains biodegradable, degradable waste. For example, non-biodegradable, you have plastic waste, right? Large amount of plastic waste is being dumped along the water. So now what happens is this entire sum of water that we get as a sewage, we can treat at least 80% of the water that is present in the sewage and use it or repurpose it for human usage, or you can just leave it to the natural water bodies so that it is safe enough for the environment as well as the or the animals that is surviving there. Now, when we talk about this water treatment, the water treatment can occur, okay, in huge tanks called as sewage treatment plants, not tanks, plants, okay, sewage treatment plants. So when we talk about sewage treatment plants, now this huge area where the water is being taken up from the municipal waste and then being recycled, it happens in two steps. One is a primary treatment, other one is a secondary water treatment. So both of them are quite different. So let's see the approaches that we do. So the crude water that is coming from the municipal waste, okay, which we call it as an effluent, okay. So now it is injected through an through a pipe and it enters into this chamber called as grit chamber. Before we let this water pass into this grit chamber, what we do is we put this physical filters. For example, if you use a mesh, now you can see that in many of the countries, especially in Thailand and or even in some parts of our country, like in Pondicherry or in Goa, what they have done is they have tried to put nets across the small stream and big streams and even some rivers. The net is tied before the water reaches into the sea. So what they're trying to do is they are controlling the flow of plastic into the oceans. They'll collect it there only without letting it to pass into the ocean because it's very hard collecting there. And then they take out all this debris. So this can be done by screening process. The same screening process can be done for the tanks also, so that the water is purified by putting a mesh. So now what happens is once you have this physical separation, now what you do is you do the grit. What is grit? Grit, it contains soil and small pebbles in the grit. So the water enters and it gets into this grit chamber. In grit chamber, what they perform is sedimentation. That means the tank is left undisturbed. Okay, the water will be flowing. Or see, when you have this water flow from higher uh, high level, I mean, like in the height, in terms of height we are talking about, this is the height of it. The water is dropped here. So when it is dropped, now what will happen? All these pebbles and small gravels, they start accumulating at the bottom. And there is constant flow of water also to the another tank. Understood? So this flow will ensure that all this debris will go and settle at the bottom. So once this is settled at the bottom, now what you get is you get two kinds of waste. I mean, two types of water. One, the flowing water, which we call it as an effluent, okay, is very, very harmful. Okay, why? Because this contains a lot of organic waste and a lot of microorganisms in it, and it needs to be treated. Okay, and the rest of water that is accumulating in the down, there is no water that is accumulating in the downward. It circulates and it goes back as an effluent only. All that is separated during the primary treatment is the pebbles, the physical debris, the plastic. All of it will get separated. Understood? So now, once we have the primary effluent, so which we call it as the sludge. Okay, so now what does it give us? So this water, which is very rich in organic waste, will be sent to your secondary treatment. Secondary treatment is biological treatment. Okay, so what happens in biological treatment? In primary effluent, though, what happened? We got all this organic rich material, and it has to go for some kind of a treatment. What is the effective way to remove all this organic waste? Can we do it physically or through any chemical process? Yes, it is possible, but unnecessarily you're spending so much energy. Why? Because you have the best biodegradable machines, that is microorganisms. I can't call it machines. Microorganisms are such effective tools to degrade anything, right? So what we do is we put this water, that is a primary effluent, into large aeration tanks. So what are aeration tanks? Usually if you see an aeration tank, see, this is the tank. Usually they have large pipes. Okay, so like they're fitted like this, and they are aerated constantly. I mean, like air is being pumped at the bottom of this tank so that the oxygen levels in this tank enriches. So when you have rich organic material and when you have oxygen, that has been given, what happens? All these aerobic microbes will start growing there. So when aerobic microorganisms grow there, what do? They feed on this organic matter. So when they feed on this organic matter, so their growth increases. Obviously, as their growth keeps on increasing and the organic material decreases, so these microbial colonies, they start accumulating and they form something called as flocks. What is flock formation? We had primary effluent. When we aerated primary effluent, huge amount of microbial growth takes place. When you have a lot of food resource and when you have good environmental conditions for you to grow, what you do reproduce. So when your population increases, now you become so much that you start appearing as flocks. You can see here, right? These are the flocks of microorganisms. So once these flocks are accumulated, if you take some amount of flock and observe it under the microscope, what do you see? You see bacterial filaments as well as the fungal filaments. Because it's not just one microorganisms, several microorganisms participate in this organic degradation process. Understood? Now, once you keep on aerating and then you have your metabolic processes that is taking place in your tanks, what happens is the amount of oxygen that is being that is present in your tanks, they decrease, right? All this oxygen is taken for the respiration, even though we aerate it. So they decrease over a period of time. Now we can reduce the amount of this waste that is present and the amount of demand that we get. What demand? Biological oxygen demand. Simple mathematics. When we have more effluents, when we have more microorganisms, when we have more effluents, we have more microorganisms, and it requires more oxygen. So that means it will take away every little bit of oxygen that is left in the water, creating this demand called as biological oxygen demand, which is a measure of dissolved oxygen. So when this decreases, it is very obvious that the effluent is too high, okay, or the organic matter in the effluent is very, very high. If the biological oxygen demand reduces, that means that, okay, our organic matter is reducing. For example, let's say we have 100 or more when we got into the tank. and after gradually it is being treated, now the BOD levels actually keep on decreasing until it comes to fair or poor. Understood? So this is called as measure of organic material or biological oxygen demand. So by definition, what do we call biological oxygen demand? It is the measure of oxygen that is dissolved in the water. So why do we have to do that? Usually whenever we have microbial growth and there is organic matter which they feed on, the respiration process demands the oxygen presence. So the use of oxygen determines how much of food they are getting. So that is a simple way to test the amount of pollutants that are present in your water or in your effluents based on the oxygen. Understood? So this is about BOD. Next one. Now, what do we do in secondary treatment? And the secondary treatment, what happens is after we keep on, uh, aerating these tanks, and then we get this flux. Okay. Now, what happens is all this effluent that is passed into the flux where the bacteria was growing, right? Now you send it to this. Sometimes the in higher water treatment plants, they're even sent into clarification tanks. Okay, so this clarification tanks, what happens is after again you have this effluent that is passed from your aeration tank to this clarification tank, you are treated with disinfectants, and you remove microorganisms from this water. For example, you do chlorination. You can leave it for sedimentation for further removal of all these unwanted things, and then you have to remove all these flocks that are produced. All these things is done in secondary clarification tank. Now what will happen? All the influent that is gotten out of this treatment is released into these water bodies, or it is reused for the public, and the rest of the waste that is collected in this clarifier, it is again returned to the sludge where it goes for further treatment. Understood? So here what is happening in the primary treatment, the removal of all this physical waste happens either through either we have this screens or screening process, or we have sedimentation or GERD chambers. Understood? In secondary treatment, what we are doing, we are trying to use microorganisms to our advantage to remove organic waste. Understood? So we have a lot of different technologies that are being introduced with respect to water treatment, and it is more effective. This is very one way of sewage treatment. We have further ways, okay, where we just aerate the tanks, and then we have spin aerators, and we have anaerobic digesters also. Understood? So this is how it is done. Now, what happens to the, where did we get these microorganisms? For example, if I have, hold on, do I have, yeah. If I have a process where I take this sewage water, and in the sewage water, I will just put it to, filtration tanks where I have remove the GERD, I remove the sediments, and then I put it for aeration tanks. In aeration tanks, what will happen? I have my microbial growth. Now, again, the fresh water comes from sedimentation tank to the aeration tank. Now I need this water to to be cleansed or to be treated as fast as possible. So if I naturally wait for the growth of microorganisms every single time, if I have to make it from the beginning, it's very time consuming. Simple, I'll give one simple example. We have milk and we need to make curd. Without the curd, it's very hard to make milk into curd, right? It takes a lot of effort. So that is what we'll do. We take some amount of curd and we put it back into the fresh milk. Okay, some amount of curd from the from the source, we take it and we put it in the milk. That will fasten the process of converting milk into curd. Similarly, whenever we have this aeration tank treatment, now what happens is after this, that when the fresh water comes, or when the fresh primary waste comes from these tanks and it is put for the aeration tanks, some amount of bacteria is introduced into this aeration tank in the form of inoculum. Okay, so this inoculum is called as activated sludge. Okay, so this is very, very important. What do activated sludge contain? Activated sludge contain the same bacterial and the fungal flocks or fungal colonies or filaments, so which were formed as flocks, correct, right? So the same little bit of flocks we use for the next round of filtration. So that is called as inocula, or it is also called as activated sludge. Understood? So when we talk about the further aspects of your water treatment, one more thing I told you is the anaerobic digesters that we use today. So anaerobic sludge digesters, what they use is, now after the aeration, and then you have your settling tanks. Now there are still some of the waste that could be removed by some of the bacteria where it involves toxic inorganic compounds. So that these organisms, they can do it in an anaerobic conditions where the bacteria and fungal sludges, they break down this inorganic molecule and produce this gas such as H2S, okay, hydrogen sulfide, methane, okay, and carbon dioxide are produced, and the same gas is being used as biogas, okay, for the public, or for example, even in some of the big hostels like medical colleges, they use this water treatment, okay, to make it more efficient. Understood? So now all this water is being treated, it will be left to the water bodies, so which is very safe. Understood? So here is a map for how the treatment of your water takes place. It's very, very important, the steps, because the steps will be asked in your examination. So we have primary effluent, and then we put it for settling tanks. Once it forms the settling tanks, we remove all the dirt and sediments, and then we leave it for aeration tanks. Okay, in aeration tanks you see this constant agitation. Constant agitation means what? Properly this water is mixed. Understood? So I told you that, right, there are several technologies that's been used. So they use this huge propellers where it spins this water and creates this constant movement in the water that leads to proper mixing of this organic matter, so bacteria can make use of it. Okay, so and then we put it for anaerobic treatment. In anaerobic treatment also, we remove all this inorganic waste, okay, and then we get the biogas out of it. So this is a map, please go through it. These are the stepwise treatment for water treatment. Okay, one of the major steps that we have taken as a country to preserve the water bodies is Ganga Action Plan and Yamuna Action Plan. Even though it is quite shameful to talk about it, our countries have the most polluted waters ever. You go to any sites where you can find a river, you find heaps of plastic, heaps of human waste, and plenty of other things. Even though we are very religiously attached, we have to take care of many other aspects as well. We have to respect the same. We have to give the same respect to nature as well. It's as simple as that. One of the sides is Ganga and Yamuna. Ganga, at least we have some progress where it is said that 2,000 crores has been spent just to clean this Ganga basin. And Ganga is quite okay. Like prior to some decades, like 1 decade, 1.5 decade back, if you would have seen Ganga, it would be really scared to see the condition of a river like that. It was that polluted. Okay, and if you see Yamuna right now, you don't think that it is Yamuna, because you see that a lot of this foam floating around in the river because of the toxic chemicals that is released. What is the reason? There are two reasons for this crisis. One is the population. The north is a very beautiful plains and there is good rivers. So what they did was a lot of population started settling around this riverbeds a long, long time ago. And the same has been continued even today. Large cities are present on the river basins only. So that led to tremendous amount of pollution of water. Second one is we have pilgrimage sites and all these human activities. Yes, it is important, but it is also important that we respect, we clean, and then we maintain properly. Now we are doing it with Ganga. It has reduced a lot. The pollution in Ganga has reduced a lot. Whereas Yamuna, it has a lot of progress has to be done. So that will happen soon. A lot of NGOs have participated. A lot of people, government and non-government people, and even the public involved in this to get the rivers cleaned up. Okay. In South, it is relatively okay. I'm not telling that we have very clean rivers, but it is okay compared to that of North. Since we have good amount of forest cover in many of the river basins, thank God, that is why our rivers did not get polluted. So, anyways, some of the action plans that we have taken up to clean the water resources or water bodies is Ganga Action Plan and Yamuna Action Plan. So here, these two were the central government projects where it was initiated to clean our rivers. Okay. So next one. Next one is we use microorganisms in biogas production. So you might have seen in the villages. If you have not seen in the villages now, even it is present now. I hope you have seen it. Okay, you can come across in villages. You used to we usually get these models that is built to encourage people to go for this biogas. Okay, production. Okay, so what happens here? A large amount of livestock cultivation. I mean, livestock we grow. I mean, like you know we raise, right? So that is why what will happen, we produce a lot of cow dung. You know Cow dung, instead of it going waste, what we do is we use cow dung to produce methane, which is a biogas. So how does it happen? You know that the ruminants or the cut chewing animals like cow, sheep, buffalo, okay, so all of them, they have this special friend which is called as methanobacterium. What it does is it helps in breaking down of this complex cell wall of the plants like cellulose. Okay, which is anaerobic bacteria. It is producing some enzymes that is helping the cow or any of these cud chewing organisms. So when it excretes, along with its dung, a lot of methanobacterium is excreted out. So what we do is we take, you see this diagram very clearly. Diagram is very, very important for both NEET as well as your exams. So we take the dung and we mix it with the water. So once it is mixed with the water, it will get washed down and it will lead to a large tank. And this big tank, we call it as a digester. So what we do with the digester is we seal this digester with a cap. Okay, it's a cylindrical cap which is hollow. Here it is hollow, it's not solid. Okay, just like this, only this much is, okay. Here it is hollow. So what happens is we seal it so that there is no air entering. So this will create an anaerobic condition, okay, like the cattle stomach. Now what will happen in anaerobic conditions, these methanobacterium, they start growing and proliferating. When they start growing and proliferating, what they do is they break down these complex sugars into simpler methane and carbon dioxide. So the methane and carbon dioxide will be, okay, will be released. Whatever it is released will be collected through a pipe, and it can directly go to the kitchen where you can use it, okay, for cooking. It's a very clean energy without any pollution. What happens to the rest of the cow dung? So the rest of the cow dung that is digested, whatever the dung that is being digested, so this is converted into a very rich sludge, which is rich in minerals, which is rich, it's like a fertilizer, biofertilizer, or manure. Sorry, not fertilizer, manure. So this rises and it will be accumulated in the sludge chamber. In the sludge chamber, what we do, we collect all this waste, that is a sludge, and we use it for a manure, okay, organic manure, so that you can grow very healthy plants, you can have healthy environment, and you can also get rid of all this organic waste without polluting. Okay, so this is about biogas plant. Okay, understood? And as this goes up, that means more gas is accumulated, and you can check it. Okay, this much gas is accumulated, and you can use it for methane. It's a very sustainable source, and it runs for a very, very longer period of time. It depends on climate also, but it is very nice. Understood? So they have taken the responsibility. Indian Agricultural Research Institute and Khadi and Village Industries Commission. Very important. These names you have to just remember. Okay. IARI, so that is Indian Agricultural Research Institute, and Khadi and Village Industries. So they develop these technologies of biogas production in India. And guess what? They are subsidizing a lot of farmers who go for this option. Even today it is open. Okay, they encourage these farmers even to practice this, so that the dependency on the fertilizers reduces and they go more of organic. So next topic is microorganisms as biocontrol agents. Okay, is it clear? Are you able to follow, guys? Are you able to listen to me? I know you can understand this topic very easily, but are all the technicalities clear? Are you able to see the PPTs? Hear the voice clearly, etc.? If not, please let me know. All right. Okay. So, using microorganisms as biocontrol agents. This is a very novel method. Why it is a novel method? Because for every single, as I told you, our ecology is very complex, and now this is always leading to interdependency. When you have fruits, when you have vegetables, when you have anything that you use, that we put it in the farm, they will attract these insects, poor insects. Okay, so what they do, they're coming for food, and we see them as pests. And again, to avoid this pest, what we do, we spray pesticides, and we consume the same pesticides. So that is the great innovative method that we are having in the agriculture right now, in the until the recent past. Now today, just by understanding the environment itself, we can use this for our advantage without having dependency on these chemicals and polluting our food, as well as uh, you know uh, brutally killing these animals with chemicals. So that is why we go for biocontrol. What happens in biocontrol? We have the maintenance of plant disease or pest infestation through microorganisms. For example, if I use ladybirds, okay, ladybirds are purposefully left on this ground, on the fields. Why? Because it controls aphids. Okay, it's biology. See, it controls aphids. Next one, dragonflies. Dragonflies control mosquitoes. Okay, so encouraging them to grow. Understood. So that is why we go for more biological approach than any chemical or industrial approach. So it's nice. So let's see how microorganisms are used as biocontrol agents. First one is Bacillus thuringiensis. You must have heard about this so much because we talked about in biotechnology. But we are not talking about biotechnology here, but we're talking about simple application of this particular bacteria. See, what it does is this Bacillus thuringiensis, even without genetic engineering, we can just put this dried spores. I take this Bacillus thuringiensis, I take the dried spores, okay, and what I do is I spray it on all the plants. For example, if I had cotton, and then the cotton was infected by this bollworm. Similarly, we have corn borer, and then we have butterfly caterpillars. So there are many diseases that are caused by these different organisms, where they are very prone to Bacillus thuringiensis. Okay, they're very vulnerable to Bacillus thuringiensis. What happens in Bacillus thuringiensis? We know that the Bacillus thuringiensis has cry protein. So when the cry protein enters the alkaline gut environment, it will destroy the epithelium lining, thereby creating the spores in the gut and thereby killing the pest. Correct, right? So that we understood in genetic engineering, but here we don't need genetic engineering. We just spray the dried spores of this Bacillus thuringiensis. We mix it in water and spray it on plants. Now, when this pest eat it, it enters into the stomach, and then the reaction happens, and the pest gets killed. Understood. So that is why BT is used as a very effective, okay, very effective biocontrol agent. Remember, you might get confused, but it is nice. You know what is more unique character about biocontrol agents? One of the most unique characteristics is, see, we spray Bacillus thuringiensis on plants. When we eat it, it doesn't infect us. Whatever it is vulnerable to, like a range of organisms, it will only affect that. It will not affect any other organisms. So this narrow range of their infectious properties is very, very beneficial. Okay, and that is why they make an effective biocontrol agents. Next one is trichoderma. So trichoderma or fungus. It's a fungus. Basically, what it does is it will form this fungal colonies in the root structures of the plants. Fungi are very, very smart actually, as their capacity to communicate with the different organisms is exceptional. So what they do is they make an association with the root system of the plants, and this mycorrhizal, fungal and root association. What it does is there are reports that apart plant the required nutrition, these fungal filaments are known to create awareness for the plants about the change in the environment. It is helping the plant to produce exact antibiotics, it will produce antibiotics to combat the diseases in the plants, and it will prevent the plant from being infected by any other pathogen. So that is the amount of association they have. They're very smart organisms, we call fungus. So they make a very deep connection with the plants, and many aspects are known about how they interact as an organism. Okay, so one of the examples of fungus being a biocontrol agent is trichoderma. Okay, so you can use trichoderma to prevent any of the plant diseases. Okay, next one we have is baculovirus. So what is baculovirus? We have another name for baculovirus that is also called as nucleopolyhedrovirus. So this nucleopolyhedrovirus, it is very vulnerable for a set of arthropods and insects. Okay, insects or arthropods. So what it does is it is very species specific. It does not affect all the organisms, and it is an effective insecticidal option or insecticide that is being developed. Okay, so this use of this baculovirus, okay, to target specific insects and to eliminate this pest problems, it is called as integrated pest management. Okay, so that is how they try to maintain this narrow spectrum. Why narrow spectrum or species specific is important? We don't want to eliminate all the insects across us. So we just want to target what we want to target so that not many of them get affected. So that is why this narrow spectrum, and then we have species specific insecticidal or biocontrol agents are very, very nice for integrated pest management. Okay. Okay. Next one is biofertilizers. Okay, use of microorganisms as biofertilizers. So we have some of the examples like Anabena, mycorrhiza, azospirillum, rhizobium, okay, so etc. So now, what are biofertilizers? It's very simple. These organisms will give us or will give our plants the required nutrition for its growth and development. Simple. Why do we spray these fertilizers? We do it because the plant is not getting sufficient nutrition. So if this is supplemented by microorganisms, why not use it? So that is why we have biofertilizers. So in biofertilizers, we can use many organisms which can give us benefits such as bacteria, fungi, cyanobacteria, etc. So first is rhizobium. What is rhizobium? It is a bacteria which will form these root colonies. Okay, what they do is they infect the roots of the plants and they start forming nodules. For example, leguminous plants. Okay, you might have heard about this legumes or rhizobium bacteria making these colonies in the leguminous plants, such as example is beans, and peas is an example for leguminous plant. So what they do is the association with them, they help in fixing the atmospheric atmospheric nitrogen. So this atmospheric nitrogen, why it has to be fixed? Because the plant's capacity to take the atmospheric oxygen and to make it into fixable forms. See, for example, to prepare amino acids, to manufacture amino acids, etc. You need nitrogen. Even nitrogen as bases require nitrogen. For all that, see, for example, we know that carbon dioxide is taken from the atmosphere and it is fixed as phosphoglyceric acid. So similarly, in plants also, they need some alternate form where they can convert this nitrogen, right? So that doesn't happen. It requires a lot of amount of energy to do that. So what does this rhizobium do? Rhizobium will make this a very simple process because it has a special enzymes that can take this atmospheric nitrogen and convert into simpler forms for the plant usage. So it's a very excellent symbiotic association. Okay, and other we have something called as free living bacterium in the soil. Okay, such as azospirillum and azobacterium. Very, very important. What is important? This term. Give examples of free living bacterium that acts as biofertilizers. Your answer should be azospirillum and azotobacter. So this was asked many times in NEET examination, the names of it and examples of it. Okay, so please write this down or just keep it in your memory. Okay, so what they do is they also help in enriching the nitrogen content in the soil. Why they ask this question is very, very important. See, we have heard about rhizobium. We know that the rhizobium makes the root, they also make the association with the roots of a leguminous plant, and it's a symbiote. We know that we have been studying for a very long time. But when it comes to free living bacteria, we ignore it. Correct, right? We don't talk about symbiosis. Whatever that is present in the, they're independent. They're left just like any other photosynthetic plants. Okay, so that is why the free living bacteria that helps in enriching the soil is Azoospermillum and Astobacter. Remember this. Next one. What is mycorrhizal association? Very excellent association. For example, you would have heard about in gymnosperms, we talk about the association of this fungi with the root system of the gymnosperm plants. So we talked about it very exclusively, how the mycorrhiza formation takes place, on all the things you might have heard about it, right? So here, what is myco? Myco means fungi, rhiza means roots. So the association of fungus with the root system is called as mycorrhizal association. Unbelievable, unbelievable benefits this fungi gives to the plant. Well, some of the experiments have suggested that the fungal association, encouraging the fungus to, infect a particular root, what it does is it will create a niche. Okay, the fungi will create this environment where it will be giving the capacity for our plants to tolerate salt content in the water, salinity stress, drought stress, whereas any other possible infections and even the minute of the micronutrients, okay, or very limited nutrients, it can derive it from the soil and it can give it to the plants. So that is why it's a very excellent association, that is mycorrhizal association. Okay, so they're symbiotic. Okay, they absorb, for example, phosphorus from the soil and they give it to the plant. They give salinity, drought, and increase the plant growth and development. Okay, next one, cyanobacteria. Cyanobacteria is also called as blue-green algae. We know that they're photosynthetic or autotrophic microorganisms. One of the major characteristics of cyanobacteria, for example, Anabera, nostoc, oscillatoria, have this specialized cell called as heterocyst. So this heterocyst, they have special enzymes that can help in fixing of atmospheric nitrogen. So this atmospheric nitrogen is used as a biofertilizer where in paddy fields, etc., you can grow this cyanobacteria, which can enrich the soil. All right, students, it's about time and exactly we have finished within time. Thank you very much. And we finish microbes in human welfare. And next one is biodiversity and conservation. Okay, hold on. Yeah, did you understand this chapter? You have any doubts with respect to any topics? Students, am I audible? Okay, clear? So if you don't have doubts, we can stop here. We can continue in the